will give us some more information on the squirrels.
It's a pleasure to be here. So I'm going to talk to you guys about risk assessments. Particularly what we're interested in is we know that animals have to acquire, process, and then respond to the information that they're receiving, both internal information and external information in their environment, abiotic and antibiotic factors. And we can see this as has been presented before in venom metering. So we know that venom metering occurs based on a number of different factors, both in predatory context and in defensive context. And so in predation, we see that satiety, so how hungry they are, venom availability, prey size, struggle intensity, prey type, all of those things affect the amount of venom that a spider or a snake or a scorpion will deliver in an injection. And we also see that, as has been presented earlier, in threat. So that's fairly well established, and so we're seeing evidence on the response side of things, how organisms respond to information. Risk assessment is really interested in how are they perceiving the threat that is approaching them in a defensive context. What information, that's generally what I'm most interested in, what our lab, Dr. Corbin in my lab is interested in is, how do animals acquire this information? What information specifically is the most important thing for them? So out of all of the sensory modalities that an organism has, whether vision or chemosensory or vibratory, which of those are they cueing in? Is everything equal, or are there some signals that are more important than others when it comes to making decisions about defensive behaviors and venom metering in particular? A little bit more, we know that there are a lot of invertebrates that acquire information, and this is a particular type of information about their environments, and we see this with navigation. So hymenopterans, ants, bees, they do something called path integration, where they are acquiring through these learning flights or learning walks information about their environment, and then they're able to use that information to respond where, in this case, they know exactly where home is relative to where they have moved, even if they take these circuitous routes. So they're holding this information within memory. And we see that scorpions actually have the potential to do this. They have all the necessary equipment to do this. So they have a lot of different sensory organs, and I'll show you a picture in an upcoming slide of some of those things. And there is initial evidence in the work of Doug Gaffin that at least Pararoctonus utaiensis is possibly doing this. And they're using their pectins as these broom-like structures to sweep the ground, and maybe they're getting this textural familiarity. So they are screening the ground, and they're remembering something about the perturbations or unevenness of the ground, and they're using that to be able to navigate back to their dens. And so here we can see some of the reasons why scorpions might make a really good organism to study some of these questions about. We see the diversity of sensory organs that they have, and a lot of them are external, right? So we've got slit sensilla, or we've got chemosensory hairs. You can see there's a pit there. And then here are the pectins. And on those pectins, there's the array of cones, and on each of these you have this battery of organs, and on them you have a whole bunch of different sensory systems there too. They are tactile sensory and chemosensory. And so they could be getting chemical information about their environment by dragging these on the ground, as well as the textural signals as well. So they make a good organism to try to study this. Unfortunately, scorpions are not well studied, or maybe fortunately for some of us who are interested in them. So we actually, if we're going to do this, we have to lay a lot of foundational groundwork, and so that's what we're going to try to do at the beginning of this study. And our lab has been working with Venomous carolinianus for a little while now, and so we've got several experiments done, and I'm going to kind of summarize this and then tie them into what we've been doing. There's actually a poster in the hallway that we left up that has some additional work and a different kind of question, but along the same lines. So the questions for this particular study was about risk assessment. This Venomous carolinianus is not well studied, there's not much data on them, and so we wanted to know, are they engaging in risk assessment? And we're going to look at two different things. Prod cycle, which I'll explain in the methods, and then the location where we touch them. And I'll also explain that in the methods. Do we see sex differences? And then we modified the environment in some way, so we modified the availability of hides. And we're wanting to see, are they learning as they move overnight, and we track their movement overnight, the amount of exploration they do, and the total amount of movement they do, and then the presence of hides, are they learning something about that that influences their defensiveness? So that's the primary question of this study. So how did we do it? Well, we ended up with 9 females and 9 males. We had a scorpion rapture, and one of them where just mysteriously a scorpion disappeared, and then unfortunately one scorpion did pass through the 3 weeks of this experiment. It wasn't looking good in week 1, and by week 2 it was gone. It was nothing that we had done, or nothing obvious that we had done. We placed them in circular arenas, and circular here is very, very important because if you know much about these organisms or just invertebrates in general, if there's a corner, they're going to find it, and then they're just going to park it and sit there for a long time. So in a circular arena, there's no place to park it, and they'll just continue to move around for hours and hours and hours overnight, walking in circles, and you'll see that in a second. We observed them overnight. We recorded, we used infrared cameras to record their behaviors, and then with the help of Doug Gaffin, we used his MATLAB script to analyze their movements. And so this generates a huge data set of about 2.6 million data points of their movement overnight for 14 hours. So include that in our model. The next morning, we would come back in and test them behaviorally to see, okay, what's going on. We poke them, and we poke them in 3 different locations: the claw, the tail, or the body. And we did this for 4 cycles. So it was always in the same order: claw, tail, body, and that's what the cycle is representing. Okay, so here is the representation of their overnight movements. You can see that some individuals are moving an awful lot. And you can see that they've moved so much that they've definitely found the hides because they've outlined them. And then some organisms are hardly moving through or exploring in the center at all, and their movement is really, really restricted to the outside. They're just walking in circles all night long. Okay? So for our results, risk assessment. Are we seeing evidence of risk assessment? Does broad cycle matter? And it does seem to matter, and this aligns with our predictions. When it comes to sting frequency, we see that initially the rule of thumb with venom use is don't use venom right away. Wait for the situation to be more threatening and then start to use it. So we don't see stinging right away, we see it escalate and then de-escalate. And the de-escalation may be related to fatigue, or it could be related to acclimation to the process. Right? 2 minutes is a long enough time for these to kind of get used to it and say, okay, this isn't as threatening as it once was. It's not as novel anymore. As far as fleeing or time spent moving around, they will respond very vigorously to run away right at first, and that declines through time. So we see a shift with cycling. As the process continues, they'll sting more and then less, and they'll flee more, and then they'll decrease in their fleeing through time. When it comes to does the prod location matter, here's where we see very, very strong results. So if you touch them on the body, the prosoma, they are by far and away stinging much, much more frequently compared to both the claws and the tail. And each of these are significant from each other. When it comes to the use of venom, so we just did this as a dichotomous variable. Do we see venom used or we do not see it? We didn't quantify the amount of venom expressed in this particular study. But we do see that venom use, when we have a noticeable bolus of venom at the tip of the stinger, is much, much more common within the prosoma than it is within the other 2 regions, or the body. And then the amount of time spent fleeing, also we see that much more commonly in the tail or the body touches than we do in the claw touches. So they will flee more vigorously as the threat increases. We predicted that the body would be the most threatening location to touch. Are there sex differences? Well, we do see that venom use is more common in females than it is in males. So when we see venom, a female is more likely. In this particular species, it's a little confounded with size, because females, these are dimorphic species, and females are much, much larger than males. And so is this size or is this sex? We have some evidence to suggest that it might be size independent. If you look at the poster again, we found that when you control for size, females are still stinging more frequently and more likely to give you venom. Here we didn't control for size, so we can't clearly say that in this particular study. In addition, we did see that hide number mattered. So when they had 4 hides available, 2 hides or 0 hides. But there was also a sex difference. So females, there's a trend of increasing that there is actually all along the way, except for hide number 2, females are greater than males. And then there's an increase. There's an increase in the 4-hide condition. They're more likely to use venom when there's more hides available. That's a little bit puzzling, and we'll return to that in a little bit. Here again, we see that males generally don't change in their sting frequency, but females are increasing in their sting frequency when there are more hides available. And we'll come back to that in a second. So what about overnight movements? Because I think we have to think not only about hide availability, but also are they aware of, is there a potential that they're aware of the hides there? So we had 2 measures of exploration. One was just total distance moved, and the other was amount of time spent in the center where the hides actually are. And when we just look at total distance moved, we see a general increase. If they move more, they're more likely to use venom. And maybe that's saying something about body condition, or maybe it's saying something about they moved more because they didn't find a hide and it's more defensive. When we consider time in the center, we actually see a negative relationship. So they are stinging less the more time they spend within the center of the arena. And then again, that's where the hides are. And to kind of drive that home a little bit, here you can see that in the red colors, that's a positive correlation. And in the blue, there's a negative correlation. And again, when you see time spent in the center, so the more time they spend in the center, the less overall movement they have. The more time they spend in the center, the less stinging they have and the less venom use they have. So this is probably our strongest line of evidence that maybe some information about where the hides are might actually be influencing them a little bit. And again, if we look at the time they spend fleeing, there's positive correlations with total movement and fleeing. So when they're behaving defensively, they're not just swapping out behaviors. It's either I flee or I sting. They're doing more of everything. They're fleeing more, they're stinging more, they're more likely to use venom, except when they have spent time in the center exploring, when the majority of the time is there. So take-home messages for this would be, do we see evidence for risk assessment in this species? Yes, I think we see very clear evidence of this in 2 ways. One, we see this change through cycle. Initially, the rule is get away from it. Get away from what's attacking you. Then I'm going to escalate in stinging, and then maybe there's a decrease through time, and that's possibly either to acclimation or fatigue. Again, with the body touches, if I'm being touched in the body, that's a much more threatening position than if I'm being touched on the claws. And so they're responding very, very differently to where you're prodding them. Are there sex differences? We didn't find as much as we thought we would find because of other experiments that we have done, but we did see some evidence for this, that females are more likely to males to use venom. Maybe that's related to size, but for some of our subsequent studies suggest maybe otherwise. And then the availability of hides. Well, it looks like there could be something, again, with this correlation matrix. There is some evidence to suggest that they're learning something about their environments. So maybe I would be tentative here. We'd like to redo this and make it more robust. But there is something there that merits further investigation. That's it. Answer some questions.